Thank you, Jean. It's always a pleasure. And now to Becky Steele, who has a scientific story guaranteed to make you think. To you, Becky.
Thank you, Steve. Stem cell therapy is rather controversial in the wider community, even though most people hardly understand the actual nature of stem cell research.
Come on, stem cells! Work your astounding scientific nonsense!
In reality, stem cells are simply cells that are pluripotent or capable of developing tissues of different cell types. Unfortunately, embryonic stem cells are harvested from fetal tissue, the root of many of its ethical conundrums.
Fetal stem cells? Aren't those controversial? In your time, yes.
However, in recent years, a method for creating pluripotent stem cells from adult tissue has been refined. These cells, called Induced Pluripotent Stem Cells, or iPSCs, are made by forcing adult cells to express certain genes that give them pluripotent properties. Because they can be made into almost any cell variety, and they are absent the ethical issues of embryonic stem cells, these iPSCs are quickly becoming pivotal in vitro genetic disorder and disease research. In 2012, Mr. James Briggs led a research team studying the progression of Down syndrome disorder caused by an extra chromosome 21. Through the use of laboratory techniques, the research team was able to determine that Down syndrome causes an excess of reactive oxygen species during development, particularly in the brain. Normally, reactive oxygen species, which are byproducts of metabolism, play an active role in keeping the body working correctly. In excess, however, the reactive oxygen species cause oxidative stress to cells, which can damage them and impede their functioning. They hypothesized that this oxidative stress was partially responsible for the deterioration of developing neural tissue, typically associated with the progression of Down syndrome. They also found that the antioxidant N-acetylcysteine, which is formed in the body as a precursor to glutathione, also an antioxidant, was able to combat the oxidative stress. This is due to the reducing properties of the thiol group on the antioxidant. The thiol group donates a proton to the reactive oxygen species, which reduces their reactivity and impedes oxidative stress. This could be likened to giving a dummy to calm a bawling baby. Induced pluripotent stem cells are becoming staple in laboratories around the world, and without a doubt, we will be hearing more of them in the future. Back to you, Steve.
Thank you, Becky. And now, a young duckling who just wanted to be a swan.